Hello, everyone, and welcome to today's poster presentation, ProDetect Rapid Assay Lateral Flow Kits for Detecting Protein Tags and Antibody Isotyping, presented by Krishna Vattem, Senior R&D Scientist, Protein and Cell Analysis, Thermo Fisher Scientific. I'm Alexis Krause of LabRoots, and I will be your moderator for today's event. We are delighted to bring in this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, please visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Krishna Vattem. I will now turn the presentation over to him.
Hello everyone, my name is Krishna Vattem. I'm a Senior R&D Scientist at Thermo Fisher Scientific. Today I'll be talking about ProDetect Rapid Assay Lateral Flow Kits for detecting protein tags and antibody isotyping. So, if you ever visited a doctor's office for a routine flu test or have used a pregnancy test kit, you must have noticed that the assays are really fast, they're accurate, and these are instantaneous basically. You walk out of the lab and then you got the result in the doctor's office. These tests are usually based on a technique called lateral flow. Scientifically speaking, they use a technique called immunochromatography. As the name suggests, it's a combination of chromatography and antigen-antibody reactions. From one's experience using these tests, one can assume that these are very rapid, sensitive, accurate, and easy to use. Now, as regards the use of this technique in life science research is concerned, these assays have very little presence. In this poster, we'll report ProDetect lateral flow assays for use in common protein expression and detection workflows, providing valuable advantages over current detection methods for rapidly monitoring expression of recombinant proteins that have a purification or detection tags on them. These assays, we hope, will address key pain points in cell culture and protein expression workflows by providing a quick, qualitative yes or no answer on the success of protein expression. If one looks at protein expression workflows, as in this figure, there are several points at which one would like to know if expression is going on according to plan or everything is going according to plan. At each step, from the point of growing the initial cultures to post-induction, one would like to know if and when the expression has started and if it has reached a plateau. Using ProDetect lateral flow assays, one can very quickly and at the point of expression, right next to the cultures where they are growing, you can monitor the expression of proteins. If proteins are secreted, all you have to do is collect the cell supernatant and dilute, if necessary, using the dilution buffer provided in the kit. And you can use a microtiter plate or a microcentrifuge tube and just dip the ProDetect strips into them and just wait for 10 to 15 minutes and you should be seeing your result. It's very fast. If the proteins are endogenous, you can use any of our lysis buffers that we provide, that we commercialize, or even if you have your own lysis buffer, that's fine too. All you have to know is the composition of the lysis buffer. And you can basically use that lysate that you make and dilute it if necessary in a microcentrifuge tube or a microtiter plate and just place the pro-detect strips into them. Just wait for 10-15 minutes and you should see a result. So as you can see in this figure, there are several points at which one would like to know whether the expression is going on according to plan or not. Not only during induction, during pre-induction, post-induction of proteins, one can also monitor expression of proteins in the elution fractions obtained from an affinity column. So you induce, you grow the cells, you induce, you lyse the cells, you take the lysate, you put it on the affinity column, you collect the fractions. Okay, you can, at the affinity fraction columns, you can quickly monitor the expression of proteins by just taking a sample of the elute, putting in a microcentrifuge tube, and just dipping the pro-detect strips into them. One can only imagine the loss of time and money if all you're doing is to just monitor the UV trace at the end of experiment and not knowing all along if you have been expressing your proteins at all. So there's a lot of value in quickly detecting the expression at each point of the expression and purification workflows. Now, the pro-detect rapid assays use either a competitive or a sandwich assay format. Competitive assays are designed to detect proteins that contain smaller tags such as his tag, HA tag, flag tag, sub tag, and so on. The sandwich assays, on the other hand, are designed to detect proteins that have larger tags on them. For example, GST, MBP, SUMO, FC regions of antibodies like human, rabbit, rat, which can also be used as tag purification tags. Very briefly, the architecture of the lateral flow strips using a competitive assay is as follows. If we look at the panels on the left side for a minute, the top half of the lateral flow strip is where you will actually see the results at positions indicated by the letters C and T. C stands for control, T stands for test line 1, test line 2, and test line 3. The bottom half is where you will have gold conjugated capture antibody conjugates, which are very specific to the analyte of interest that you're trying to detect. And the bottom half is also the region where you will actually dip this strip, the lateral flow strip, into the sample. As I said before, you can have a sample in a microtiter plate or a microcentrifuge tube, or any other of your convenience, any other device of your convenience. Coming back to the top half, a control antibody is immobilized at the control line indicated by the letter C, which will appear both in positive and negative results. This is just to say that yes, the chromatography is working as planned. The liquids are flowing from bottom to the top. The target analyte is immobilized on the membrane as 3 distinct test lines, as I said before, T1, T2, and T3. These are the analytes actually impregnated into the membrane. Now, I'll come to the actual how the results are looking part in a minute, but this kind of format is only specific to the competition assays. You'll have a control line and you have 3 test lines. Well, how do the results look at the end of about 10 to 15 minutes of incubation of this lateral flow strip into solution? In a negative result where no analyte tag protein is present in your sample, or the concentration of the tag protein in a sample is well below the detection levels, the gold conjugated capture antibodies, which are in the bottom half of the strip, will actually migrate to the top and will bind to the analyte positions, T1, T2, T3, which will result in the formation of 3 red lines. So in a sample which does not have the analyte, you will see the test lines lighting up. In a positive test, on the other hand, where the sample contains the target tag proteins, the gold conjugated antibodies will bind to the tagged proteins in the sample instead now, and therefore will not bind to the test lines upon the top. As a result, the test lines do not appear. So, very briefly, If a sample has an analyte, the test lines will not show up. The sample does not have the analyte, the test lines will show up. As the concentration of the tagged protein in a sample increases, the number of test lines will gradually decrease and eventually disappear. So the concentration of the tagged proteins is inversely related to the number of test lines appearing on the strip. Now, moving to the sandwich assays. So sorry, I was just going to go back to the previous slide. If you look at on the right side, we were trying, this is just an example to show that the sample which contains a MIG-tag protein, the test lines actually disappeared. If there is no MIG-tag protein, the 3 test lines appear. This is to the right side of the slide when you see. Sorry, coming back to the sandwich assays. In a sandwich lateral flow assay, the gold conjugated capture antibodies, which are present at the bottom half of the lateral flow strip, are very specific to the target analytes, and these are embedded in that region. Okay? And additionally, there is one more antibody called the detection antibody, which is present again, which is also specific to the analyte, but at a different epitope. So in this sandwich assay conditions, you have 2 antibodies. One is gold conjugated and impregnated into the conjugate pad, and the other antibody is impregnated at the test line. So both the antibodies are specific to the same analyte, but raised in different epitopes. So upon application of the sample that has analyte present, the analyte of interest is present in the sample, the capture antibodies bound to any present analyte in the sample travel along the length of the membrane and are resolved as 2 lines, test line and the control line. And both these lines start lighting up. This is exactly like a pregnancy test kit, where one line showing up means you do not have a positive result. If 2 lines show up, that means it's a positive result. The sandwich assays exactly work like a pregnancy test kit. If, as I said before, if there are no bands displayed at the test line, that means the sample is negative. So the control lines here do appear both in a positive and a negative assay, indicating that the assay is working as planned. So the sandwich assay is very easily to relate to a common assay out there in the market, either in the doctor's office or in the pregnancy test kit, as I said before. If we look at the example on the right, we try to detect GST tag protein in the sample, and the 2 strips that are present there. In a negative sample, which is indicated as none, there is no GST in the sample, only 1 line shows up. In the positive result, where GST is present in the sample, 2 lines are lighting up. One is at the control line, other at the test line. So these assays are extremely simple to read and to analyze. Just remember, sandwich assays, presence of line at the test line indicates the sample is positive for the analyte. A competition assay, on the other hand, disappearance of the test line indicates the presence of the sample, the analyte in the sample, sorry. Now, we have a number of kits in the market. So these are either broadly categorized into a competitive or a sandwich assay. As I said before, sandwich assays are designed to detect larger tag like a SUMO, GST, MBP, and the FC fragments of human, rabbit, and mouse. The competitive assays, on the other hand, designed to detect smaller tags, HA, stub tag to AV tag, MIC, V5. We have one more kit. It's called a dual tag detecting kit. Detects both the flag and the HES tag. But this one works in a sandwich format. So you have to have a dual tag on the protein with HES and HA on either end or at the same end, doesn't matter. But the assay works in a sandwich format where appearance of the result at the test line indicates success of presence of analyte in the sample, success of presence of dual tag in the sample. Okay, but if you look at just the flag or just the HES, just individually, they work in a competitive format. Again, talking about the range of detection, ideal range of detections, sandwich assays are very sensitive. I mean, I love sandwich assays because they are very sensitive. They can detect, like, the ideal range is 0.1 to 10 microgram per ml that we recommend, where the signals are really strong. But you can also get signal where the concentration of the protein is, for example, 10 nanogram per ml instead of 100 nanogram per ml as indicated here. You can also see 10 nanogram per ml detection, and you can also see proteins which are in the range of 100 microgram per ml. But the signals are slightly weaker, so that's why we recommend being 0.1 to 10. But if you're outside the ranges, that is also fine. The signals are slightly weaker. In a computation assay, on the other hand, ideal range of detection is 4 to 20 micrograms per ml. Anything above 20 micrograms per ml, the signal just is disappeared, completely disappeared, which means there is no upper limit for a computation assay. Anything beyond 20 micrograms per ml, the signal is gone, indicates the presence of the protein in your sample anyways, do not worry about it. Anything below 4 micrograms per ml, sometimes it's really hard to say, but I would not recommend somebody going below that microgram per ml for detecting smaller tag proteins. So between 4 and 20 micrograms per ml, it's a nice gradient of disappearance of the three test lines. Each of the test line will have different concentration of the analyte in it as a background. But as you go from 4 to 20 micrograms per ml, gradually each of the line disappears, and beyond 20 micrograms per ml, all the three test lines disappear. So next slide. The next two slides, we're going to show two application data using our ProDetect kits. In this slide, which you're looking at it right now, we compared the sensitivities of a chromatin stainable stain gel versus a Western blot versus our ProDetect GST assay strips. As you can clearly see, our ProDetect strips are detecting proteins as little as 0.1 microgram per ml in this example here. But as I said before, even if you go 0.01 microgram per ml, you will still see a signal. So these are awesome. These are extremely sensitive sandwich assay kits, down to 10 nanograms per ml or 5 nanograms per ml, even in some cases. You can clearly see that when you do a chromatin stain, everybody knows you got to get a stain, you got to de-stain. It takes anywhere from 2 to 4 to overnight. If you do a Western blot, the same issue. You got to go grab the antibodies, you got to do the Western, you got to run the gel, transfer, primary, secondary, wash, you got to do all these steps. It takes anywhere from 4, if you're fast enough, to overnight. But ProDetect strips, 15 minutes max, and you should see a result. It's extremely fast and accurate. Now, we're not showing a ton of data that we are generated here. It includes specificity of these strips, how specific these are, and how accurate these are in terms of cross-reactivity or in terms of seeing any other background proteins from lysates. They are very clean, these strips. These are very specific. All the data is posted on our website, or you can also see some of the data posted in our IBs. This is one more example here how we use the ProDetect kits to just look at whether we have successfully expressed a protein or not. This is an example. As I said before, there are two ways of looking at expressed protein in cells. One is if it is secreted, no problem. Just take a supernatant, dilute it in the ProDetect assay dilution buffer, and you just dip the strip into that, and you should get your result in 15 minutes. The protein is endogenous, which is most of the cases in this case too, as well as the one that you're seeing right here. We expressed a dual tag protein that contains flag and a His, and this was expressed in a bacterial cell. The protein is inside the cell. We had to quickly take about 100 microliters of the cell and lyse for about 5 minutes, take the supernatant, and then quickly dilute in our dilution buffer. We took about a microliter of the lysate or about 5 microliters of the lysate to detect the expression of the dual tag protein. As you can clearly see, in the 3 panels that you're seeing right there, the left panel is the dual tag protein detection kit for flag and His, which works like a sandwich assay. You can clearly see at 1 and 5 microliters, you can see the signal showing up. In the middle panel, which is specific for the flag, you can see that 1 in 5 microliters, the signal disappears, which is also indicating the presence of the protein. In the HES alone, strips, which is on the right side, you can clearly see at 1 in 5 microliters, the signal also disappears. Okay, so you can use these kits very rapidly at the point of need, either in the cell culture room, next to the wherever you're trying to express your protein. And not just that, as I said in the beginning slide, you can take your elution fractions coming out of an affinity column and quickly look at whether your protein is being eluted or not, whether the peaks that are coming out of the UV trace are really your peaks, the protein that contain the protein of your interest. So one thing that you had to also pay attention is the background of these lysine buffers. We provided extensive information on what are the limits of detergents that you can use in your system. Ideally, if you dilute your sample, all the effects most likely disappear. Anything that is coming from a high SDS or high urea or whatever be the high detergent conditions, these effects will really minimize if you dilute your sample. If you do not dilute the sample, you have to know the limits of how much is allowable, and that's published in our instruction booklet as well. So, in conclusion, ProDetect rapid assay kits are really quick, easy, sensitive assays that can be used to analyze the progress and progression of protein expression and purification. The ProDetect rapid assays can assist in decisions for go, no-go at every step of the workflow. You don't have to waste hours and days of time, and then at the end of the day, you'll say, oh my God, I didn't express the protein, what I was doing all these days. You don't have to go through those kinds of pain, And these assays are compatible with a lot of commonly used component lysis and buffer solutions. As I said before, all you have to do is what is exactly the composition of your lysis buffer. And as I said before, these assays have been also characterized and made sure that the background from the bacterial yeast or mammalian cell lysates do not interfere with these assays. So in short, these are extremely powerful and rapid assays, and please definitely you should try these assays and let me know if you have any questions. I would love to answer any question that you have. Thank you so much for your time.
Thank you, Dr. Vattem, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what is the minimum volume of the sample you need for this assay? And can we add sample directly to the strip, or does the sample need to be in in a microplate?
Yeah, so the minimum volume I would recommend is 100 microliters. I mean, you can go down to 75 microliters, but 100 microliters is an ideal volume to do the assay. You don't have to do this in a microplate. You can have your samples in any test tube, in a clean test tube or a microcentrifuge tube. I think the other question is, can we add sample directly to the strip, or does it have to be in a microplate? Yeah. I mean, I know people who have been using the traditional the lateral flow devices are used to adding sample directly to that cassette-based situation, where the strip is actually inside a plastic cassette. The cassette casing is not there in our case. So what you can do is you can have a clean surface, for example, like a parafilm or something, or a plastic surface, and then you can lay your strip on top of it, and then you can add in increments of 25 microliters and up to maximum 100 microliters. You should see a result just fine.
So, Dr. Vattem, is this assay compatible with urea or other denaturing agents such as SDS?
So we posted a list of compatible reagents and their maximum allowable concentrations that could potentially interfere with our assays. For example, like SDS, as the question is asking. There is a certain recommendation to be within certain limits, something like around 0.1% SDS. I don't exactly remember, but even urea, there is a certain concentration. We also posted concentrations for detergents like other detergents like Triton, I believe, and also salt concentrations, how far you can go on salt, sodium chloride, and yeah, I mean there are a bunch of reagents that are commonly used in protein extraction buffers. We listed them, and we said you got to be within these concentrations to have a non-interfering, so that these concentrations do not interfere the assays. And I mean, as I said in the presentation, if you dilute your sample about 10-fold, most of the effects go away. So you don't have to worry about, oh, what's there in my buffer, because a lot of times these reagents are proprietary. If you're buying for a company, you don't even know. They won't even tell you exactly what's there in the lysine buffer. So I would just dilute 10-fold and make sure it's roughly in the concentration that is recommended that I was mentioning earlier, the detection limits, and you'll be fine.
All right, so our next question is, these assays seem to be only qualitative. Any plans to get them to quantify proteins of interest?
Yeah, you're right. These are purely qualitative, yes or no. At this point, we do not have a quantitation-based assays, but that certainly is of interest to us. We are looking into those assays as we speak. But these are designed to be very extremely cheap and so once you talk quantitation, you have to buy instrumentation and you have to generate some kind of a standard curve, and it becomes more complex. So the idea of these assays is extremely fast, yes or no, go to the next step. And that's where this fits in.
And it looks like our next question is, why does the testban signal get weaker as the concentration of sample increase as in figure 4?
Sometimes what happens is if we have excess of analyte present in your sample, all of it cannot be conjugated with the gold conjugated antibody present in the conjugation pad, which is the bottom of the strip. So what happens is the cold antibody, I should call it cold because it is not, the cold antigen, which is not actually conjugated to the antibody, migrates, and then it blocks all the test signals on the T line. So if your concentration is in the right range of the analyte is in the right range, most of the analyte will be bound by the antibody, which is conjugated to gold, and these will ideally get arrested at the test line. You should see a signal. So this is something like a hook effect. Higher concentrations usually give out weaker signals. So in those conditions, I would recommend doing just one more dilution of your sample, about 10-fold, 100-fold, and do one more time the same assay. If the assay signal completely disappears, which means there's not enough antigen, but if the assay signal improves upon further dilution, which means there's a lot of protein present in your sample.
So it looks like we have a time for just a couple more questions.
Yes.
The next question is, what about fusion proteins? Is there any problem with these?
If I understand right, fusion proteins, fusion to a tag? I mean, I'm not exactly following the question here. When you say fusion proteins, here's the thing. As long as the tag present on your sample. So remember, these assays are designed to detect tags on the protein. These tags, as I listed in one of those slides, can be purification or detection tags. The assays that we develop these for include HES, HA, FLAG, MIC, V5, Strep Tag 2, GST, SUMO, MBP, and these tags. As long as these tags are available, it doesn't matter what else is there in your protein. and you should be able to detect these tags in your proteins, which means you are indirectly detecting that, yes, you expressed your protein if you detect these tags which are present on your protein.
So Dr. Vattem, this is going to be our last question. What's the sensitivity of the assay for this tag compared to Western blot?
These are very sensitive. As I showed in one of the experiments, you can detect in a his tag. Again, it depends. When it's a Western blot, it depends on what antibody you're talking about. Some antibodies are really good. Some antibodies are not that good. And of course, Western blot takes about an overnight time, basically, right, to do the entire process. You need to have equipment and everything. These assays are designed to be very simple. As regards sensitivity is concerned, you can detect a concentration in the range of 10 microgram per ml of his tag protein in your sample very easily. Anything about 10 micrograms per ml, you should be very comfortable if you're in that range. So if in a Western blot, you're talking about 10 microgram per ml sample on a Western, probably you can get a signal with a 10 microgram per ml his tag protein in your sample. Again, depends on the antibody. But I should say, for any competitions in assays in general that are small tags, is HA flag, so on and so forth, if you're in the range of a 10 microgram per ml, you should be able to detect your protein very easily. That should not be a problem. Again, the concentration that I'm referring to is a concentration of actually the tagged protein in your sample. Your sample could be in the midst of a ton of other lysate proteins. It doesn't matter. But your protein, the tagged protein should be in that range of concentration, which is 10 microgram per ml.
I would like to once again thank Dr. Vattem for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share that with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.